Look closely at a damp, barren rock face, and you will likely find bryophytes. They are universally known as the amphibians of the plant kingdom. They earn this title because they live a paradoxical existence. They are terrestrial plants anchored in the soil, yet they remain biologically tethered to water in order to complete sexual reproduction. In the evolutionary timeline, bryophytes serve as the physical and biological bridge. They sit directly between simple aquatic algae and the tall, highly complex vascular plants that dominate most landmasses today. But to understand how they survive on land, we have to look at what they lack. Bryophytes possess no true roots, no true stems, and no true leaves. This structural deficit exists because they completely lack vascular tissues. They have no xylem, the internal tissue that transports water, and no phloem, the tissue that distributes nutrients. Without an internal plumbing system to move resources against gravity, bryophytes were forced to engineer alternative structural and reproductive models to survive outside the water. Let's look at exactly how they do it. This wireframe demonstrates the foundational plant body of the bryophyte, known as the thallus. A thallus is an undifferentiated, flattened structure that replaces the need for true stems and leaves. To anchor this thallus to the ground without a deep root system, the plant relies on downward-growing filaments called rhizoids. Rhizoids do not actively pump water like standard roots. Instead, as the soil dampens, they absorb water purely through passive capillary action, the physical tendency of liquids to draw upward through narrow spaces without external force. On a cellular level, the thallus is composed of eukaryotic autotrophs. This means their cells contain a distinct nucleus, and they manufacture their own food using chlorophyll A and B. The surface-level cells are packed with multiple chloroplasts, allowing the plant to maximize photosynthesis across its flattened exterior. Because there is no internal xylem or phloem, resource distribution is intensely localized. Every individual cell must absorb water and nutrients directly from its immediate environment through osmosis, the movement of water molecules across a cell membrane. Advanced angiosperms, or flowering plants, use deep roots and stiff vascular columns to grow vertically and defy gravity. The bryophyte cannot. The prostrate thallus architecture is an optimized engineering solution. By flattening out and staying close to the damp substrate, it guarantees immediate surface water absorption, bypassing the need for an energy-intensive plumbing system. Bryophytes employ a reproductive strategy known as the alternation of generations. This is a life cycle that continuously alternates between a haploid phase, which carries a single set of chromosomes, and a diploid phase, which carries two. In this diagram, we see the dominant, free-living, and photosynthetic phase, the gametophyte. As labeled, the gametophyte is haploid. When the plant is ready to reproduce, multicellular, jacketed sex organs develop directly on the surface of this green gametophyte. Here we see the male antheridium and the female archegonium. The precise function of the antheridium is to produce and release thousands of motile antherozoids. These are sperm cells equipped with two whip-like tails, or flagella, for propulsion. Meanwhile, the female organ, the archegonium, is shaped like a flask and holds a single stationary egg. This is where the amphibious requirement comes into play. The antherozoids cannot move across dry land. They require a physical, continuous film of external water to mechanically swim from the antheridium to the archegonium. When an antherozoid reaches the egg, fertilization occurs inside the archegonium. The fusion of these two haploid cells creates a diploid zygote, now carrying two complete sets of chromosomes. In many lower organisms, this zygote would immediately divide to have its chromosome count. The bryophyte diverges here. The zygote rapidly divides via mitosis, a process that duplicates the entire genetic code to build a multicellular body directly on top of the original gametophyte. This newly constructed, distinct diploid structure is called the sporophyte. The sporophyte is organized into three specific anatomical zones: the foot, which anchors it, the seta, which acts as a stalk, and the capsule at the apex. This arrangement requires a parasitic anatomy. The sporophyte is not a free-living plant. It cannot perform photosynthesis, so it remains permanently tethered to the green gametophyte to extract its nutrients in water. Inside the mature capsule, the plant finally executes meiosis, a specialized cell division that halves the chromosome count back to a single set. This process produces thousands of haploid spores. When the capsule bursts, wind disperses these haploid spores across the soil. If a spore lands in a suitably damp location, it germinates and grows into a new independent gametophyte, completing the cycle. Bryophytes can also bypass this sexual cycle entirely. They perform asexual reproduction either by fragmenting their thallus or by shedding gemmae. Gemmae are specialized multicellular green buds that detach and immediately clone the parent plant. The mechanics of the haplodiplotic cycle reveal a striking reality. A mature moss is two genetically distinct organisms stacked together. The green base feeds and protects the parasitic stalk, ensuring the vulnerable terrestrial embryo survives until it can release spores. Botanists divide the bryophytes into two primary classes based on their structural complexity: Hepaticopsida, known as liverworts, and Bryopsida, the mosses. Liverworts, like the genus Marchantia, shown on the left, represent the simpler architecture. They develop a strictly dorsiventral thallus, meaning it has distinct upper and lower surfaces, that grows flat and closely hugs the ground. Because of their strict adherence to the substrate, liverworts rely heavily on small receptacles called gemmae cups to rapidly clone themselves across the immediate surface area. Mosses, such as the genus Funaria, possess a slightly more complex architecture. They utilize a two-stage development process for their gametophyte. When a moss spore germinates, it enters the protonema stage. It grows into a creeping, green, filamentous web that spreads horizontally across the soil. From this horizontal web, the plant enters the leafy stage. Lateral buds emerge from the protonema and grow upward into slender, vertical axes covered in spirally arranged leaves. These upright shoots eventually carry the sex organs. Both classes share a complete lack of vascular tissue, but while liverworts remain strictly prostrate, the mosses engineered a secondary growth stage that introduces vertical orientation to an otherwise flat existence. When we zoom out from their cellular mechanics, we see the true ecological scale of what this architecture accomplishes. Operating alongside lichens, bryophytes function as ultimate pioneer colonizers. They are uniquely equipped to settle on harsh, barren rock surfaces where nothing else can survive. As they spread, they execute petogenesis, the biological and chemical creation of soil. They secrete acids that chemically degrade the rock, while their rhizoids physically wedge into microfissures, breaking the stone down into mineral dust. As the bryophytes decompose, they add organic matter to this dust, forming a nascent layer of soil. The resulting dense moss mats then act as physical barriers, trapping moisture and preventing heavy rainwater from washing the new soil away. Beyond building ecosystems, specific bryophytes have immense commercial value. The most notable example is the moss genus Sphagnum. Over centuries, dead sphagnum slowly compresses in acidic, waterlogged bogs to form peat. For generations, humans have actively cut and extracted these dense, dark blocks of peat to burn as a highly combustible fuel source. Sphagnum also possesses an exceptional sponge-like cellular structure. It can hold up to 20 times its own dry weight in water. This extreme moisture retention makes it the premier packaging material for the agricultural industry, used globally for the transshipment of living plants, flowers, and biological materials. Bryophytes are highly specialized survivors. Their unique, rootless architecture successfully bridged the gap between the ocean and the land. By breaking down solid rock, they physically manufactured the soil that allowed all subsequent terrestrial ecosystems to take root and thrive.